Hello, my name is Maruk and this is our channel InformationBoxTicket Lifestyles, where we delve into the latest scientific discoveries and their implication. And we are discussing groundbreaking research on the Epstein-Barr Virus, or EBV, and how new approaches might help us combat its associated diseases. Let's dive in. Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes. Exactly 60 years ago, pathologist Anthony Epstein and virologist Epstein-Barr discovered a virus that would carry their names, Epstein-Barr Virus. The Epstein-Barr virus, a member of herpes family, was the first proven virus to cause cancer in humans. Today, Epstein-Barr virus is incredibly common. About 90% of the adults are carriers of the virus, and around 50% are infected before the age of 5. While many remain asymptomatic, acute infection can lead to glandular fever, also known as kissing disease. Beyond causing glandular fever, Epstein-Barr virus has cancerogenic properties and is suspected to contribute to autoimmune diseases like multiple sclerosis. Unfortunately, no drugs or vaccines are currently available to specifically target Epstein-Barr virus within the body. Researchers from the University of Basel and the University Hospital Basel have made a significant strides in understanding Epstein-Barr virus. Led by Professor Christopher Hess, the team discovered the Epstein-Barr virus programs immune cells called B cells by increasing the production of an enzyme known as IDO1. This enzyme, IDO1, ramps up energy production in infected cells, allowing them to proliferate rapidly. This process is crucial for the virus to establish a chronic infection and potentially lead to disease such as cancer. In patients who have undergone organ transplant, the immune system is suppressed to prevent organ rejection. This suppression can allow Epstein-Barr virus to cause blood cancer, known as post-transplant lymphoma. Researchers found that IDO1 is upregulated months before this cancer is diagnosed, suggesting it would serve as an early biomarker. Interestingly, IDO1 inhibitors were previously developed to treat established cancer but were unsuccessful. This research indicates these inhibitors could be repurposed to dampen Epstein-Barr virus infection and prevent Epstein-Barr virus-associated diseases. Experiments in mice showed that IDO1 inhibition reduced B cell transformation, viral load, and lymphoma development. This discovery opened up the door to new preventive strategies and treatments of Epstein-Barr virus-related conditions, particularly in vulnerable populations like transplant patients. To summarize, the reprogramming of B cells by Epstein-Barr virus through the enzyme IDO1 presents a promising target for intervention. The potential repurposing of IDO1 inhibitors could revolutionize how we approach Epstein-Barr virus-associated diseases. We will be following this research closely and keep you updated on any new discoveries. Thank you for watching. If you find this video informative, please like, share, and subscribe to stay updated on this latest science and medicine. See you next time on InformationBoxTicket Lifestyles.